Hello, this is going to be an overview of a number of videos in a playlist which reviews how respiratory systems evolved and how the human respiratory system works. First off, the earliest animals didn't have respiratory systems. These flatworms don't have respiratory systems. They're doing all of their gas exchange through their skin. This is the reason why they are so flat. In fact, there are fossils from the Ediacaran period which are 3 and even 6 feet long, but perhaps as thick as cardboard. And so it seems that while animals didn't start with respiratory systems, there were ways around this. They could breathe through their skin, and a flat shape with a lot of surface area was one of the ways around this. When we look at the earliest deuterostomes, these are the animals which include the vertebrates and the echinoderms. One of the first things that the deuterostomes, such as the modern hemichordates, had was they had a modification of their throat region, of their pharynx, so that they could take in water through their mouths, and then it came out these slits in their throat. And so here are modern hemichordates. These are primitive deuterostomes. Like the early deuterostomes, they had slits in their throat. This allowed them to take water in their mouths, and it came out their throats. And this was a very effective way of feeding, as I'll mention. Now, this is fascinating simply because in addition to feeding, which was the original purpose of this. So here's a hemichordate looking like one of the Cambrian fossils, which could, suck something, creating suction. Water comes in the mouth, it comes out the slits in the throat, and that was a large object. They probably would have been feeding on smaller objects because the throat would be lined with mucus, and it would trap small particles, and cilia would sweep this towards the stomach. We'll get back to that. But chordates, as we'll see in a second, would simply then add blood vessels to these slits. And so that now, in addition to water passing out here, the blood vessels would absorb oxygen. And so therefore, this then became gills. If you look at a shark's throat, notice that it has prominent blood vessels, branches of the aorta, which goes to each of these pharyngeal slits. This is a component of its gills. Now, while this is fascinating, this is actually a feature of all of the vertebrates. So here you see a pig embryo, and notice it has slits in its throat. Human embryos have these pharyngeal slits in their throats. So this is a feature of all of the chordates, even if we have modified it and no longer use them as gills. Once again, the original purpose was that food particles would get caught in mucus that could then be swept towards the throat and swallowed into the stomach for food. Interestingly, we still do this. We still catch particles in the mucus which lines us, and cilia sweeps this towards the stomach. But we humans, this isn't a way that we feed. Rather, this is a way of cleaning our lungs and preventing the particles which we ingest get into our lungs. So the very first deuterostomes developed these slits in their throat. And while it was primarily a feeding mechanism for these organisms, later chordates, such as the lancelets alive today, they're invertebrates which are closely related to the vertebrates. They then send blood vessels to these pharyngeal arches, which then made them function as gills. And whether you're talking about an invertebrate lancelet or a fish, this is how the circulatory system is set up. A ventral heart sends blood through an ascending aorta. And then it forms these aortic arches. We number them. Lancelets have many, but by the time we get to fish, there are 6. So the deoxygenated blood is sent to these slits in the throat. Here, capillaries exchange gases, and then the red oxygenated blood then joins a descending aorta, which then distributes oxygenated blood throughout the body. Not only is that how fish organize their blood vessels and breathe using gills, but that is how these invertebrate chordates began. Interestingly, human embryos, we have this same setup. We have a ventral aorta, which sends aortic arches to these throat regions, but then we reorganize our throat when we realize that day in embryonic development that we no longer want to develop gills. And so it was in the invertebrate chordates that not only did gills develop, the same type of gills that fish would use, but the circulation to these gills, where deoxygenated blood is sent to gill filaments, capillaries there exchange gases, and then oxygenated blood is then sent throughout the body. Now, one other comment is that in these gills, there is an interesting setup where water and blood flow in opposite directions. This is what is known as countercurrent flow, when you have two fluids flowing in opposite directions. So in this animation, the blood, which is going to be coming from this direction and going this way. Oops, sorry. So the blood that was going from right to left started blue. You know, it started deoxygenated, and it would leave then the gills oxygenated. But the water was going in the opposite direction. And there's a great advantage to that, because the water, as it moved from left to right, it went from, you know, oxygenated, I've colored that red, to deoxygenated, I've colored that blue. So notice, this is where the water coming in, you know, beginning in the gills is most oxygenated. And here is where it will be least oxygenated. That's why I will pigment it blue. Here we have the blood, which is least oxygenated. It has just been sent from the heart. And here is where it will be most oxygenated. Because these two fluids are flowing in opposite directions, here, the water, even though it is at a low oxygenation, still has more oxygen in the blood. So oxygen goes into the blood. Here, the blood is more oxygenated, but this is where the water is the most oxygenated. So here, oxygen continues to go from the water into the blood. So because of this countercurrent flow, oxygen is entering the blood all throughout the gills. If the two flowed in the same direction, oxygen would come into the blood, and then they would get to a point where they equilibrated, and then there would no longer be any more oxygen. So this countercurrent flow allows the blood leaving the gills to be more oxygenated than it otherwise would. So that's essentially how gills work. And the homologue of gills evolved before there were fish. Fish simply adapted something that was already present in invertebrate chordates, which preceded them. Now in fish, the gills are protected by opercular bones and are internal, whereas they can be external as they are in amphibians. Amphibians like tadpoles can have internal gills, but also salamander larvae and the newborn tadpoles can have external gills. External gills work just by absorbing oxygen from the surrounding water. The problem is if the water is stagnant. All right, if the water isn't moving, then once oxygen has been absorbed, there's not as much oxygen. And so this would then limit the amount of oxygen which could be absorbed. So salamander larvae have external gills. Tadpoles in their first day of life have external gills, but then they can also have internal gills while they are in their aquatic stage. These are tadpoles which hatch from eggs, being more elongated and having these external gills, as you can see. So that's great. However, oxygen can still be scarce, especially in waters which are stagnant. And so therefore, just because a fish has gills doesn't mean it's getting enough oxygen. And one of the significant things is the swim bladder, which evolved to supplement this. Now, it's helpful to remember the history of fish. The first fish are known from the early Cambrian. They were only a couple of inches long, maybe an inch or two, and they were, those primitive jawless fish. The first jawed fish occur somewhere between the Ordovician and the Silurian, and by the end of the Silurian, we have the first bony fish. The earliest bony fish form a number of groups which are largely extinct today. So the teleost fish, which represent the majority of fish alive today, they first evolved in the Mesozoic era. But we do have a couple of representatives from Paleozoic the ray-finned fish, and this would include things like sturgeons and gars. One of the exciting things about sturgeon, gars, and bowfins, another primitive actinopterygian fish, is the swim bladder. Notice how big it is. This would stretch the entire length of the ventral body cavity, starting in the esophagus near the mouth, and then going down into the region of the pelvic fin. So this is something that the earliest bony fish had, this very prominent swim bladder. Modern teleost fish, they use this to primarily for buoyancy, and that's why they can navigate so well. But the primitive bony fish, like this sturgeon, like the gar, and so here's a gar, and like bowfins, they're not using it for buoyancy the way teleost fish do. Instead, they use it to breathe. All right, so that would mean they would have to go to the surface, as many fish do, and they take a gulp of air. Why? They have gills. Yes, but the gills may not be getting enough oxygen for these fish to be as active as they'd like, especially in still water. And so the earliest bony fish had two potential sources of oxygen: gills, which could extract it from the water. And unfortunately for fish, there's not a whole lot of oxygen in the water. Oxygen is a non-polar molecule. Water is polar, and so there's not a whole lot of oxygen which dissolves in water. Here you see the inside of a swim bladder. Notice that it is divided as lungs are. Notice that swim bladders empty into the esophagus, as does the lungs of amphibians. So for example, salamander lungs empty into the esophagus. And notice in the swim bladder that red blood cells are coming really close to the atmospheric air. The wall between them is very, very thin. That's essentially the same setup as one would see in lungs. See here, the swim bladder, there's the blood, here's the air, very thin membrane. Lungs will be the same way. All right. So the first bony fish had a swim bladder whose primary purpose, was to take in oxygen from the air. Some of these fish can drown if you prevent them from breathing the air. They don't get enough from their gills. Sarcopterygian fish, they have the same essential structure, but they've done two things. They've split it into two, and they've increased the blood vessels which go to it. Now, we no longer call this a swim bladder, we actually call it lungs. So lungfish have lungs. Now, we're not being generous and kind and saying, oh, let's call them lungs. No, those are lungs. And they actually work better than amphibian lungs. I mean, some amphibians have given up on their lungs and just breathe through their skin. And so lungs evolved from a swim bladder in fish before fish came out onto land. While most fish like that perch or this moray eel are using the swim bladder primarily for buoyancy, the ability to take in air from oxygen from the surrounding air, that was one of its first functions. So let me just hit home on that point a couple of additional times. So here you see catfish coming on land to feed, and they can breathe while they do it. Here you see fish coming up and gulping air, right? And so there are a bunch of fish which can actually take oxygen out of the air, and they have modified a number of their the parts of their anatomy to do so. Lungfish are as significant in that they actually have true lungs. So here you see a lungfish. It has gills, but after a certain point, that's not enough oxygen. So there it goes. It's taking a gulp of air, which is going to now bring into its paired lungs, unlike the single swim bladder. And then it's now going to be getting its oxygen both from the oxygen passing over its gills and the oxygen in its lungs. And so when we get to tetrapods, the realization is that as they adapted for life on land, their fish ancestors had already achieved some of the necessary steps. Fish already had lungs before tetrapod amphibians evolved. And the lungfish, sarcopterygian fish, are remnants of this. So in the Devonian, there was a series of sarcopterygian fish groups, which gradually became more and more like the amphibians in a number of features. But in this chapter, we're obviously focusing on the respiratory system. So there are lots of advantages for these fish. They could supplement oxygen, especially in stagnant water. If they now had bones in their fins to let them move on land a little bit, they could leave a pond which was drying up and then find a pond that would allow them to survive. They could escape predators. The biggest predators were in the water at the time. They could find food on land. The first insects were wingless and would have been easier to catch. And so there were lots of advantages to being able to adapt to land. One could lay eggs in a vernal pool on land where there wouldn't be any predators which would prey on these young. And so it was from these the modified swim bladder that tetrapod lungs evolved. Now, the main thing about lungs is, once again, that very thin epithelial lining which separates the blood and the air. So here's a cross-section of lungs, and one can see that there's air in these air sacs. These are known as alveoli. But notice how thin the lining is. Alveoli are lined by simple squamous epithelia. This is the thinnest type of cell in the human body. And so blood and air do not mix. All right, air does not go into blood and blood does not mix with the air. But because the air sac, the alveolus, is lined by the simple squamous epithelia, the thinnest layer of cells. And because blood vessels, capillaries, are lined by the thinnest possible cells. So here's a lining of a blood vessel. See how thin that endothelial cell is. It is simple squamous epithelia. Look at this cell, its flat nucleus. Look how thin they are. So the endothelia which line blood vessels are simple squamous epithelia. And so therefore, here's air, here's blood. They don't mix, but they come so close to each other because the lining of the air sac is simple squamous epithelia. The lining of the blood capillary is simple squamous epithelia. And because of that, these two become so close to each other that oxygen can go from the air sac into the blood and carbon dioxide can go from the blood into the air sac. And so this is essentially how lungs work. This alveolus is the functional unit of a lung. Each lung has about 150 million of these. Now, those type 1 cells, those thin cells, they are the main cells of lungs. There are a couple of other cell types, certainly immune cells to help protect the lungs, but there's also what's called the type 2 cell, which secretes a solution called surfactant. Let me explain. If an alveolus wasn't kept moist, the cells would dry out and die. But keeping an alveolus moist is a problem because that causes the alveolus to constrict. Water has a surface tension, which would cause the alveolus to shrink. And actually, this is the cause of respiratory distress syndrome, which takes the lives of tens of thousands of infants per year, where they can't breathe when their alveolus collapses. This is especially a risk in premature infants. To prevent that, there's this other cell type, the type 2 cells, which secrete something which isn't water. It's a protein mix known as surfactants. And so thus the alveolar fluid has less surface tension than pure water would, and these alveoli would stay open. Now, not only is that important for lung function, but the reason that I'm speaking from home right now is because I'm making this video during the coronavirus self-isolation. And from what I have read, the type 2 cells of the lungs are some of the primary cells that the coronavirus infects. So just an added point. How does air come in and out of the alveoli? Well, there's a law in physics known as Boyles' law, where there is an inverse relationship between volume and pressure. If pressure is measured by the number of collisions against the walls of a container, say here, the pressure would go up. There would be more collisions when the volume goes down. So as the pressure goes up as the volume goes down. The same thing here. If you now make the volume bigger, the pressure will go down. There will be fewer collisions against the walls of the container. And this then brings us to a very important point in vertebrate or tetrapod breathing. You are not a salamander. Now, that probably is important on a number of fronts, but look at this one. Look at how the salamander is using its throat muscles to bring air in and out of its lungs. That's exhausting. I can tell because I was filming the salamander who would rather run away from the biologist with a camera in its face, but it couldn't because it couldn't get enough oxygen to its muscles. And it just had to, pause here. And so notice that its ribs aren't moving. See, the sides aren't moving. It's drawing oxygen in and out of its lungs by sucking it in and spitting it out. Now that isn't working well for the salamander, and it certainly wouldn't work for us. And so the amniotes, the reptiles, birds, and mammals, they developed a new innovation. They lengthened their ribs, and the ribs then contact the sternum, the breastbone. But because of this, the muscles between the ribs, known as intercostals, there are muscles between ribs, obviously. If you ever eat ribs and you're eating ribs and you're wondering, what am I eating? You're eating the muscles between the ribs, which are known as intercostal muscles, literally between the ribs. So when amniotes develop these longer ribs, amphibian ribs tend to be short. Now the muscles which move the ribs now change the volume of the thorax, which was important in breathing. Mammals did one more. Mammals developed a diaphragm, all right, a muscle which now separates the heart and the lungs from the organs of the ventral body cavity. And this is something that makes mammals unique. So just to back up for a second, if you were to ever stand in front of a mirror and you let your guts hang out, you suck them in, you let your guts hang out, you suck them in. The reason you can do that is that you've lost ribs in your lower back. You don't have lumbar ribs. Reptiles did, and mammals lost them. Mammals lost lumbar ribs so that their guts could hang out. Why would mammals do that? Well, because when the diaphragm pushes down on the guts, which makes the guts hang out, this then makes the lungs longer, all right? And so when we inhale, the way that we inhale is we have our external intercostals contract, right? So the muscles between the ribs on the outside, they contract and they bring the rib cage out and up. They make the rib cage longer this way. And then the diaphragm pushes down. That makes the thoracic cavity longer that way. And so you can make your thoracic volume increase both this way, when the external intercostals bring your rib cage out and up, and also this way when your guts hang out and your lungs get longer. Either way, the volume goes up. When the volume goes up, the pressure goes down. And if you watch me, if I make my chest bigger, this is uncomfortable. If I open my mouth, the air goes rushing in for free. The salamander had to suck it in. But all I have to do is to make my thorax bigger and the air will go in for free. And so the external intercostals, which the amniotes developed, and the diaphragm, which the mammals developed, they make the thoracic cavity bigger, and this then results in an inhale. So when you're at rest, you are inhaling when the external intercostals and the diaphragm muscles contract. So a resting inhale relies on external intercostals and the diaphragm. Now, when we're breathing at exercise and inhaling more forcefully, then some neck muscles can become involved in, well, like the scalenes and the sternocleidomastoid. They can give a little tug on the rib cage from above. So a resting inhale has the external intercostals and diaphragms contract. A forceful inhale, the diaphragm, external intercostals, and some neck muscles contract. When we exhale at rest, all we have to have is the external intercostals and the diaphragm relax. So the only muscles needed for the a normal exhale is that our inspiratory muscles relax. If my external intercostals and my diaphragm relax, my chest gets smaller. Now, I can make my chest even smaller because internal intercostals can make the rib cage even smaller than it is at rest, and abdominal muscles can suck the guts in. And so in labored breathing during exercise, to exhale forcefully, we now have internal intercostals and abdominal muscles contract. A resting exhale doesn't have any muscles contract. We just have our inspiratory muscles relax. So that is how gases come in and out of the lungs, and that then is most of the respiratory system. Just a few final notes on other aspects of respiratory anatomy in vertebrates and their evolution. Fish began with two nostrils. If you were to look on a shark, there are two nostrils. But what the Sarcopterygian fish did was they moved one of the nostril, the posterior nostril, to the jaw margin and then to the roof of the mouth, right? So for a shark, water comes in here, water leaves here, and they're trying to smell for blood, in the water, for example. But once the nostril moved to the palate, now a fish with its mouth closed could bring oxygen in this anterior nostril, and the oxygen could now, keep going in through the palate to the lung. In the nasal cavity, there are these plates which stick out, known as conchae, the ethmoid skull bone, and then another bone called the turbinate. These form these three pairs of plates, so that when we inhale, this air doesn't come straight into our lungs where we have our most delicate tissue, that simple squamous epithelia. Instead, these conchae cause turbulence in the air. It swirls around, and this will warm the air if it's cold. This will moisten the air if it's dry. Our very delicate lungs, would be disturbed if they were breathing cold, dry air. But also then if there's any germs or dust or mucus, this can get caught germs or dust or dirt or cigarette ash or etc. This can get caught in the mucus, which is then swept towards the throat and swallowed into the stomach. So once again, we're using that same mucociliary response, which the early hemichordates did. We're not using it to feed so much as to bind this debris and mucus and bring it away from our lungs, given that our lungs are made of that simple squamous epithelia, which is so delicate. So in our nasal cavity, we have those conchae. We have two organs for smell, the main olfactory epithelium and the vomeronasal organ, although in the higher primates like ourselves, this doesn't work. And the nasal cavity is separated from the oral cavity because we mammals have a palate, because we need to breathe so much. We've separated this so we can have food in our mouths and still be breathing. That second nostril is here, the internal nares. That allows the air that comes in the external nares to keep on going towards the lungs. From the nasal cavity, air then goes into the pharynx, and we split the pharynx into three regions. There's a nasopharynx behind the nasal cavity. There's an oropharynx behind the oral cavity, and there's a laryngopharynx above the larynx. As embryos, this is a fascinating area because we form that throat with slits, just like all chordates do. But as adults, there's not that much that goes on in the pharynx. We have 5 masses of tonsils there, and the opening to the middle ear, called the pharyngotympanic tube, is there as well. That helps to balance air pressure on either side of the eardrum. Now, the next region is the larynx. The fish have all these cartilage bars for the pharyngeal arches. We take this cartilage and we fuse a number of the pieces to form this one single structure called the larynx that has a number of different cartilages in it, like the epiglottis. The epiglottis saves our lives every time we swallow food because there is a shared passage for food and air. Air goes from the nasopharynx to the oropharynx to the laryngopharynx to the larynx and trachea. Food goes from the oral cavity through the oropharynx, the laryngopharynx to the esophagus. So note that there's this shared region for both food and air. But if food were to go into the wrong tube, if food were to end up in the larynx and trachea, it could block an airway and we could suffocate. And so the epiglottis is an elastic cartilage which closes over the glottis. This is the opening of the larynx, so that food has no choice. It goes into the esophagus because going into the airway simply isn't an option. The larynx has a number of cartilages. The vocal cords are located here as well. From the larynx, food goes into the trachea, which is maybe 10, 16 inches long, and has these C-shaped rings of cartilage which keep it open. The trachea branches into 2 primary bronchi, which then branch into secondary bronchi and tertiary bronchi, and smaller and smaller respiratory passageways until one gets to the bronchioles. Once you get to the bronchioles, there's no longer any cartilage in the walls of these respiratory passageways, and they have smooth muscle, which will determine whether they are open. So just like with blood vessels, we can do vasoconstriction and vasodilation. With respiratory passageways, we can do bronchoconstriction and bronchodilation. Unfortunately for asthmatics, because there's no cartilage, if one were to continually inflame the bronchioles and their walls were to get too thick, now when they do bronchoconstriction, that could actually then seal off the airway and stop breathing. From the bronchioles, we then have the air sacs, those alveoli where gas exchange occurs. Around the lungs, we have a serous membrane known as the pleura. So just as there is a serous membrane around the heart, known as the pericardium, which has an outer and inner layer and a lubricating fluid in the space between, the same too with the lungs. So if you look here at these cat lungs, you'll see the cat lungs are continually moving as air goes in and out. But we certainly don't want blisters to develop. And so we have a membrane around the lungs, a pleural membrane that has a lubricating pleural fluid in the pleural cavity between. Now, this space needs to have a pressure which is just a little bit less than atmospheric pressure. That helps the alveoli to stay inflated. If that pressure were not negative, if it weren't less than atmospheric pressure, and this would say occur if you say punctured the pleural cavity, then if atmospheric air can get incorporated into this space, that would cause a lung to collapse. The lungs originate as a single bag in lungfish or in amphibians, but we mammals, we have separate lobes of our lung. And so one could discuss lung anatomy. Lungs are roughly triangular with a base and an apex, and then they can be divided by fissures into different lobes. We have one more lobe on the right side than on the left because we have one more fissure there. And the left lung has a space here for the heart. The heart angles to the left of the midline. 2/3 of it is to the left of the midline. And so therefore there is a cardiac notch in the left lung. So when studying the respiratory system, that was a little bit about the evolution of the respiratory system. We can see that our respiratory structures evolved over stages, and so we start with the hemichordates having some of the features of fish gills before there were fish, and then there was the swim bladder and then the lungs in fish before there was life on land. Intercostals were added later. The diaphragm was added later. And so you could look at the various stages of the human evolution through ancestors and determine which aspects of the respiratory system evolved at each step. There are other topics which one could discuss in the respiratory system, such as the gas exchanges which are occurring. So in the lungs, what we call external respiration, oxygen is going from the air sac into the blood, all right? So we breathe in oxygen, and here in the capillary, it's going into the blood while carbon dioxide leaves. So in the lungs, we see external respiration as oxygen goes into the blood and carbon dioxide leaves. And so I have a number of videos that go into this in greater detail. When we send blood to our tissues, I'm sorry, should have more videos here. When we send blood to our tissues, that's what's known as internal respiration. And in internal respiration, now the oxygen is leaving the plasma going to the surrounding tissue, and carbon dioxide enters the blood. All right, so we have two places of gas exchange. In the lungs, where we practice external respiration, oxygen goes into the blood and carbon dioxide leaves. And then in the tissues, where we see internal respiration, carbon dioxide is entering the blood and the oxygen is leaving. Now, these gases don't dissolve well in the polar water of plasma, so they have to be carried. So most of the oxygen in your blood is being carried by the protein hemoglobin. This is in red blood cells. Hemoglobin can also transport some carbon dioxide, but a lot of carbon dioxide then travels as the form bicarbonate. So when water and carbon dioxide fuse, it forms carbonic acid, which forms bicarbonate. And bicarbonate is the form in which 70% of the carbon dioxide traveling through your blood is traveling. So this has been a quick overview of the evolution of respiratory systems and how the human respiratory system works.